Grab the green ice bucket and in a drawer nearby, get the key labeled ice. Go downstairs to room 37 and in the back left corner there will be an ice machine. Fill up the bucket with ice. Find a 14 milliliter Falcon tube. Calculate the appropriate amounts of each ingredient to add to your lysis buffer based on how many samples you will be extracting. Find the STE and add the appropriate amount to the Falcon tube. In the left-hand freezer, find the box labeled Proteinase K and RNase A. Inside, there should be tubes with black dots. That means these are the open ones. Take those out. Thaw the solution by warming it in your hand. It needs to be thawed so that you can fully mix it. When the RNase A and the Proteinase K have thawed, mix them by vortexing. Keep the enzymes on ice until you've added the appropriate amounts, then put them back in the freezer. Find the last ingredient, the 20% SDS, and add the appropriate amount to the buffer. When you've added all the ingredients, put the cap on the tube. You need to press down firmly until it clicks to know that it's really on. Then, mix the lysis buffer. Find a tube rack. In the drawer beneath the centrifuge, get as many centrifuge tubes as you need and put them in the rack. Set up your lab notebook page now. Put the date, the title, and make 2 columns, one for the sample name and one for the sample weight in grams. Get out 2 weigh boats and put one on the scale. Tear the scale to zero it out. Find a razor blade and carefully remove the safety covering. Bring a pair of forceps over to the work area. Fill a foam cooler with 2 of the ice packs from the green freezer. Find the blue plastic pestles in the cabinet. Make sure you have everything assembled before you go get the frozen spleens to keep them frozen as long as possible. Get the orange gloves and the key marked -80 freezer. Bring the white cooler with the ice packs. Go down to room 35, put on an orange heat glove, and open the freezer. Move over the white foam pieces. Find the rack that you're looking for and take out the box of samples. Make sure that you put the rack back in and close the freezer back up while you're working. Transfer as many samples as you want from the box into the white cooler. Close up the box and be sure to put it back in the freezer. Lock the freezer before you leave. Look inside the cooler and record all the sample names. Label the tubes accordingly before you take the spleens out. Take out the first sample and quickly unwrap it. Using the forceps, grab the spleen and put it on the weigh boat on the scale. Get the weight of the spleen and record it. Also record the sample number. Pick up the spleen and place it onto the other weigh boat. Use the razor blade to cut off a small piece. Verify that the piece weighs approximately 10 milligrams. Wrap the sample back up in the foil it came in and put it into the white foam cooler to keep it frozen. Label one of the centrifuge tubes with the sample number and place the small piece of spleen inside. Use one of the blue pestles to crush the spleen against the side of the centrifuge tube. It's kind of difficult and annoying, but do your best. Add 700 microliters of lysis buffer to the tube. Make sure that the spleen is floating in the buffer and not stuck up against the wall. Make sure the caps are on tightly on all of the tubes and move them into the center of the rack. Secure them with 2 pieces of tape so that they don't fall out while they're in the shaker. Wedge the rack in between the pieces of metal in the bottom of the shaker so that it doesn't fall out. Make sure the temperature is at 37 and the safety is off, then turn it on. Turn off the shaker and take out the rack containing your samples. Turn on the light in the hood and take out the phenol from the right-hand fridge. Put it in the hood so it warms to room temperature. Add the appropriate amount of phenol. To draw it up correctly, press down on the pipette tip through the aqueous layer and get the organic layer. Add the phenol to the Falcon tube and then dispose of the tip in the bag. Find the CVAG mixture and add the appropriate amount to the tube containing the phenol. Tightly cap the tube and invert it to mix. Set it aside so that the layers can separate. After a few minutes, you'll see an aqueous layer on top and an organic layer on the bottom. Get rid of the aqueous layer and dispose of it in the phenol chloroform waste jar. Open your samples carefully to avoid contaminating the inner cap. Add 700 microliters of the phenol and CVAG mixture to each of your samples. Emulsify the solution by inverting and vortexing 3 to 5 times. Make sure the cap is on tightly, and if you detect that any samples are leaking, transfer the solution into a new tube and repeat the process. Open the lid to the centrifuge and take out the plastic inner lid. Load your samples in directly across from each other so that the weights are balanced and the centrifuge does not break. Put on the plastic lid, close the lid, and set the timer for 5 minutes. Listen for a few seconds to make sure it's spinning properly. The speed should be maximum. While the centrifuge is running, you can get out more centrifuge tubes and start labeling them. When the light goes off, it's safe to open the lid. Remove your samples carefully, trying not to jostle them because you want the aqueous and organic phase to stay separated. This is what your sample should look like: organic on the bottom, protein interface in the middle, and the aqueous layer, what you want to keep, on top. Place a sample between your thumb and index finger and place the empty tube between your other fingers. Using an L200, carefully suck up the aqueous layer. You want to try not to get any of the protein. If you keep pulling up the protein, try pipetting out the entire protein interface first, then take out the aqueous layer into another tube and perform another phenol chloroform extraction before moving on to the next step. This is what your aqueous layer should look like on the left, and on the right is the organic waste layer. Pour out your organic waste layer into the phenol chloroform waste jar, and then dispose of the centrifuge tube in the plastic bag. Estimate the volume of aqueous layer you have, then add an equal volume of CVAG to your tube. Like before, you want to make an emulsion, so repeat the process of inverting, shaking, and vortexing until you create a uniform emulsion that should look something like this. You may have tubes with different volumes, so make sure to balance them correctly. If you can't find a match with an equal volume for one of your tubes, use the water balances that should be nearby the centrifuge. Find the appropriate amount that will counterbalance the weight. Remove your samples from the centrifuge and bring them to the hood. They should look like this. If you get any samples with a protein interface, remove the aqueous layer and extract again. As before, carefully pipette up the aqueous layer. You won't see a visible protein interface during this step, but you can still tell the difference between the organic and aqueous phases. Be extra careful not to get any protein during this step because you need to have a very pure DNA sample for the end product. Find the 5 molar NaCl. Based on the volume of your sample, add the appropriate amount of NaCl. This will help precipitate the DNA. Cap your sample and mix it until you don't see any more Schlieren lines. Get out the bottle of ethanol from the flammable storage cabinet beneath the hood. Estimate the amount of aqueous sample you recovered and add twice that volume of 100% ethanol to the tube. As you mix your tube, you should see white, stringy DNA start to precipitate out. You have to centrifuge the DNA so that it will form a pellet on the bottom of the tube. Place the tubes in the centrifuge with the cap front facing outward. This will put the pellet in the correct position. Run the centrifuge for at least 10 minutes. This is what the pelleted DNA should look like. Carefully remove the supernatant and don't remove the pellet. This is why it was important to keep the cap fronts facing out during centrifugation. Place the supernatant from each sample into its own individual labeled tube. In case the pellet doesn't precipitate, you can try adding more salt to the supernatant. Find the 70% ethanol and add 500 microliters to each of your samples to wash the DNA pellet. Then remove as much of the 70% ethanol as you can and dispose of it. Turn on the right heat block to the low setting so it preheats. Load your samples as before and pulse the centrifuge by hitting the momentary button to get the liquid down from the walls. Remove the residual ethanol. Get some millipore water and add 5 microliters of water to each of your samples to rehydrate the pellet while the ethanol evaporates. Put the samples on the heat block with their caps off for 10 minutes. When time is up, check that all the ethanol has evaporated by wafting the scent towards you. Find the 1/10 TE and add 50 microliters to each of your samples. If the pellet had formed on the wall, make sure it's pushed down fully into the TE so it dissolves. Vortex each of the samples.